Hold a single chlorophyll molecule up under enough magnification to see its structure, and at the exact center of that structure sits one atom. Not carbon, not nitrogen, not oxygen, though all of those are present in the surrounding ring. Magnesium. One atom of magnesium, anchored at the core of a porphyrin ring, is the structural foundation that makes the entire molecule capable of doing the one thing every green plant on Earth depends on for survival: capturing a photon of light and converting it into chemical energy. Remove that single magnesium atom, and the molecule that remains, called pheophytin, cannot perform photosynthesis. It is structurally almost identical to chlorophyll. It is biologically useless without that one atom at the center. This is not a minor biochemical footnote. It is the reason an entire category of plants suffering, one of the most commonly misdiagnosed problems in home gardens, traces back to a single missing mineral that costs less than $2 a box at any pharmacy in the country. This video is about what happens, mechanically and visibly, when a plant runs short on magnesium, how to recognize that specific deficiency with confidence rather than guesswork, the documented method for correcting it, and the diagnostic mistake that causes a significant number of well-intentioned gardeners to apply the wrong fix to the wrong problem. Here is where the mechanism becomes visible above ground, and where most home diagnosis goes wrong. A plant experiencing magnesium deficiency does not simply turn uniformly yellow. The pattern is specific, and recognizing that specific pattern is the entire key to accurate diagnosis. Magnesium is what plant physiologists classify as a mobile nutrient, meaning the plant has the biological capacity to relocate it from older tissue to newer, more metabolically active tissue when supply runs short. When magnesium becomes scarce in the soil, the plant makes a triage decision. It pulls magnesium out of its oldest, most established leaves and redirects it toward new growth at the growing tips, where the demand for active photosynthesis is highest. This is not random plant behavior. It is a documented, predictable resource allocation strategy, and it produces an equally predictable visual signature. The oldest leaves, typically the lowest leaves on the plant, begin showing yellowing first. But the yellowing does not spread evenly across the leaf surface. It appears specifically between the veins, while the veins themselves remain distinctly green for a noticeably longer period. Plant pathologists call this pattern interveinal chlorosis, and it is, structurally, the visible signature of magnesium leaving leaf tissue exactly where chlorophyll production is failing, while the vascular tissue, which has different cellular composition and different magnesium requirements, holds its green color longer. A tomato or pepper plant, photographed at this stage, typically shows a striking marbled pattern on the lower leaves, dark green veins running through pale yellow tissue, almost like a stained glass window, while the newer growth at the top of the plant still looks entirely healthy. That contrast between healthy new growth and yellowing old growth is itself diagnostic information, because it tells you the plant still has functioning uptake and translocation systems. It is not dying. It is redistributing a resource it does not have enough of. Here is the detail that separates an accurate magnesium diagnosis from a wasted bag of Epsom salt. And it deserves to be stated plainly because this single point of confusion accounts for a meaningful share of why home remedies for yellow leaves fail. Multiple nutrient deficiencies produce yellow leaves. They do not produce the same pattern, and the differences are specific enough to diagnose correctly without sending a soil sample to a lab. Nitrogen deficiency also targets older leaves first, Because nitrogen is likewise a mobile nutrient, the plant relocates toward new growth under scarcity. But nitrogen deficiency produces uniform yellowing across the entire leaf, veins included. There is no green venation pattern, no marbled contrast. The whole leaf simply fades evenly toward pale yellow and eventually brown. Iron deficiency produces a visually similar interveinal pattern to magnesium deficiency, green veins against yellow tissue. But iron is an immobile nutrient inside the plant. The plant cannot relocate it from old tissue to new. As a result, iron deficiency shows up first and most severely in the newest leaves at the growing tips, the exact opposite location from where magnesium deficiency appears. A plant with yellow new growth and healthy green old growth is showing an iron problem. A plant with yellow old growth and healthy green new growth is showing a magnesium problem. That single distinction, which end of the plant is affected first, is the most reliable field diagnostic available to a home gardener without laboratory equipment. Overwatering and root rot produce a different signature entirely, typically a more uniform wilting and yellowing across the whole plant simultaneously, often accompanied by softness at the base of the stem or a sour smell from the soil, rather than the clean interveinal pattern that signals a specific mineral shortage. Misreading any of these patterns and reaching for the wrong amendment does not just fail to help. In the case of repeated Epsom salt application to a plant that is actually nitrogen or iron deficient, it can introduce excess sulfate into the soil without addressing the actual shortage, occasionally compounding nutrient imbalance rather than resolving it. Diagnosis before treatment is not a cautious suggestion here. It is the difference between a fix that works in days and a guess that wastes a season. Once interveinal chlorosis on older leaves has been confirmed as the pattern present, here is the documented mechanism behind why Epsom salt, magnesium sulfate, corrects it as quickly as it typically does. Magnesium sulfate is highly water-soluble, considerably more so than many of the magnesium-containing minerals naturally present in unamended garden soil, which means that once dissolved and applied to the root zone, the magnesium ions are immediately available for root hair absorption, rather than requiring weeks of microbial or chemical breakdown to become plant accessible. This solubility is the entire reason the correction timeline for genuine magnesium deficiency is measured in days rather than the weeks typically required for slower release amendments. Once absorbed, magnesium travels through the plant's vascular system to the chloroplasts, where it becomes available to insert into newly synthesized porphyrin rings, restoring the structural foundation that allows new chlorophyll molecules to form correctly. Existing yellowed leaf tissue, where the chlorophyll breakdown has already occurred, will generally not regreen substantially, since that magnesium has already been cannibalized and relocated. What gardeners typically observe instead is that the yellowing on affected leaves stabilizes rather than worsening, while new growth coming in afterward shows markedly improved, deeper green coloration, since that new tissue now has adequate magnesium available during its initial chlorophyll synthesis. This is an important expectation to set clearly. The fix is forward-looking rather than retroactive. It stops the problem from progressing and corrects it in subsequent growth, rather than restoring already damaged tissue to its original color. The application method matters as much as the diagnosis. The standard, well-documented ratio is 1 tablespoon of Epsom salt dissolved completely into 1 gallon of water, applied as a soil drench directly around the root zone of the affected plant, repeated every 2 to 4 weeks during active growing season for plants showing confirmed magnesium deficiency. Complete dissolution matters considerably more than it might initially seem. Undissolved granules sitting on the soil surface release their magnesium unevenly and slowly, while a properly dissolved solution delivers a uniform, immediately available dose directly to the root zone where uptake actually occurs. A foliar application, diluted at a lighter concentration and misted directly onto leaf surfaces, can supplement the soil drench and is sometimes used for a faster, visible response on severely affected plants. Since some absorption occurs directly through leaf stomata, though the soil drench remains the primary and more reliable delivery method for sustained correction. Tomatoes, peppers, and roses are the three garden plants most frequently and most visibly affected by magnesium deficiency, largely because all three are particularly heavy feeders during their fruiting or blooming phases, periods of intense metabolic demand when magnesium consumption for chlorophyll production and enzyme activation runs highest. Tomatoes, specifically, tend to show this pattern reliably starting in mid to late summer, once the plant has shifted substantial energy toward fruit production and the demand on every mobile nutrient, magnesium included, intensifies considerably. This seasonal timing is itself a useful diagnostic clue. Interveinal yellowing on lower tomato leaves that appears specifically as the plant moves into heavy fruit set, rather than earlier in the season during primarily vegetative growth, fits the magnesium deficiency pattern with particularly high reliability. There is a secondary mechanism worth understanding as well, because it explains why the response to correctly applied magnesium sulfate often appears faster and more complete than the chlorophyll mechanism alone would predict. Magnesium also functions as a critical enzyme activator inside plant cells, required for the proper function of numerous enzymes involved in ATP production, the cellular energy currency every metabolic process in the plant depends on, and in the activation of rubisco, the enzyme responsible for the actual carbon fixation step of photosynthesis. A magnesium-deficient plant is therefore not only short on the structural component of chlorophyll, it is operating with reduced enzymatic efficiency across multiple metabolic pathways simultaneously. Correcting the magnesium shortage restores both the structural and the enzymatic dimensions of the deficiency at once, which is part of why the visible improvement in new growth often arrives faster than a purely structural explanation alone would suggest. For anyone standing in a garden right now looking at lower leaves with that distinctive marbled, green-veined, yellow tissue pattern, while the top of the plant still looks perfectly healthy, the diagnosis is specific enough to act on with confidence, rather than guesswork. Confirm the pattern. Confirm the location, old leaves rather than new growth. Dissolve 1 tablespoon of Epsom salt completely into 1 gallon of water. Apply it as a soil drench at the base of the plant. Watch the new growth that follows, rather than expecting the already yellowed leaves to reverse. The chemistry at the center of every chlorophyll molecule on the planet has been understood for over a century. The application of that chemistry, for a home gardener standing over a single struggling tomato plant, costs less than the price of a cup of coffee and works on a timeline measured in days. If this kind of precise, mechanism-first gardening knowledge is what brought you to Rooted Grace, the like button and the subscribe bell keep this work going every week. Drop a comment below describing exactly where the yellowing appears on your plant, upper leaves or lower leaves, veins green or veins yellow too, and I will help you work through whether magnesium is actually the deficiency in play or whether something else deserves a closer look. The next video in this series covers the second most commonly misdiagnosed deficiency pattern in home vegetable gardens, one that looks deceptively similar to this one, but requires an entirely different mineral to correct. That one is coming next week. Until then, look closely at which leaves are yellowing before reaching for any remedy. The pattern is always trying to tell you something specific.